Hey, how's it going, everyone? This is FixerMed, and welcome back to my high-yield biochemistry review series for the USMLE Step 1, NBME CBSE, and NBME CAS examinations. This will be part 9 of a multi-part video series detailing the entire discipline of biochemistry in a high-yield but comprehensive manner. So without further ado, let's go ahead and get started on today's content. Today we'll be mainly focusing on glucose-6-phosphate dehydrogenase deficiency, also known as G6PD deficiency. Now, the main highlight of G6PD deficiency is that it's part of the hexose monophosphate shunt, known as the HMP shunt, a pathway in the cell that produces NADPH. And since NADPH is a crucial molecule for reducing agents and is particularly important in protecting cells from oxidative stress, this can result in hemolysis. Now, I'm going to go through some specific facts regarding the epidemiology, the inheritance, the triggers, and the diagnostics, alongside the treatments and preventive measures. So let's go ahead and get started. So for epidemiology, G6PD deficiency is one of the most common enzyme deficiencies globally. It affects males more frequently than females, as the gene responsible is located on the X chromosome. Is more prevalent in certain populations, including those of African, Mediterranean, and Southeast Asian descent. The main inheritance pattern of G6PD deficiency is an X-linked recessive genetic disorder. Males are more commonly affected, as stated previously, while females can be carriers or, in rare cases, manifest symptoms if they inherit both of the defective genes from both parents on their X chromosomes. The main triggers of G6PD deficiency are certain drugs like antimalarials, like primaquine, sulfa drugs, dapsone, and certain antibiotics, which can trigger hemolysis in individuals with G6PD deficiency. Certain infections or illnesses can also act as triggers. Foods like fava beans are known to trigger hemolysis in certain cases. For diagnostics, diagnosis is often confirmed through blood tests, including G6PD enzyme activity assays, hemolysis markers such as Heinz bodies, and bite cells on blood smear may be observed. For treatments, there is no specific cure for G6PD deficiency. Management involves avoiding triggers that can induce hemolysis, such as specific medications and certain foods. In severe cases of hemolysis, supportive care and blood transfusions may be required. For preventive measures, genetic counseling for families with a history of G6PD deficiency is crucial to understand the risk of passing the condition to future generations. Awareness of triggers and avoidance of substances that can induce hemolysis is also a great preventive measure to potentially stop G6PD deficiency from manifesting in certain patient groups. Now on the next slide, I just have all the text that I spoke to you guys, so be sure to take a look at that and write notes on it if necessary. Otherwise, I'm going to be moving on to the NBME application-based reasoning questions. So we have a certain researcher studying the influenza virus and the phenomenon of antigenic shift. Which of the following mechanisms best explains antigenic shift leading to pandemics? This isn't tied explicitly to G6PD deficiency. I just wanted to warm you guys up with an easy question. Alright, I think I gave you guys enough time. If you need more time, pause the video now. Moving on. So antigenic shift, which leads to pandemics, involves the reassortment of viral segments, particularly those coding for hemagglutinin and neuraminidase. Therefore, the correct answer in this question is reassortment, and we'll go through and see why the other answer choices are incorrect. A. DNA recombination. Influenza is an RNA virus, and reassortment involves the exchange of RNA segments, not DNA recombination. B. Gene mutation. Antigenic drift, not shift, is associated with point mutations in hemagglutinin and neuraminidase, leading to epidemics. D. RNA splicing. Reassortment involves the exchange of entire RNA segments, not RNA splicing. E. Transposon insertion. Transposons are not involved in the process of antigenic shift. Now, guys, this isn't exactly a biochem question. I just saw it pop up in a third-party QBank, and I thought it'd be a good review. When I'm doing the high-yield genetics series, we'll be talking more about these, so don't worry if you can't recall these, but these also tie into biochem in certain situations, so just keep that in mind. Next question. We have a 13-year-old male that presents with yellow eyes following treatment for infection. A blood smear shows precipitated hemoglobin within red blood cells. What is the most likely mechanism for this condition? You guys a few seconds to think that through. All right, I think that's enough time. If you need more time, pause the video. I'm moving on. The presented symptoms and blood smear findings are indicative of glucose-6-phosphate dehydrogenase G6PD deficiency, leading to increased RBC membrane oxidation. G6PD deficiency results in deficient NADPH production via the HMP shunt, and NADPH is crucial for reducing agents which protect red blood cell membranes. Therefore, the correct answer here is C, increased red blood cell membrane oxidation. The incorrect choices can be explained as follows. A, decreased red blood cell membrane permeability. G6PD deficiency does not directly affect membrane permeability. B, impaired oxygen binding to hemoglobin. The symptoms are more consistent with hemolysis due to membrane oxidation, not impaired oxygen binding. D, enhanced production of heme. G6PD deficiency does not lead to increased heme production. E, accelerated red blood cell production. The issue in G6PD deficiency is related to the increased susceptibility of existing red blood cells to oxidative damage, not the rate of production. So that's why those answer choices are incorrect. Let's move on to the next question. We have a 19-year-old female that presents with enzyme deficiency and increased indirect bilirubin. Which enzyme is most likely deficient in this patient? All right, I think I gave you guys enough time. If you need more time, pause the video. Moving on now. Pyruvate kinase deficiency leads to impaired ATP production in red blood cells, preventing the normal functioning of the sodium-potassium pump. This results in the inability to pump sodium out of the cell, leading to water retention, red blood cell swelling, and eventual lysis. Increased indirect bilirubin is a consequence of hemolysis. Therefore, the correct answer is pyruvate kinase C in this question. Let's go through and break down why the other answer choices are incorrect. A, G6PD deficiency causes hemolysis, but does not involve sodium pump dysfunction and water retention. B, hexokinase deficiency primarily affects the early steps of glycolysis and is not associated with the sodium-potassium pump dysfunction seen in this case. D, catalase. Catalase is involved in the breakdown of hydrogen peroxide, not in glycolysis or the sodium-potassium pump. E, aldolase. Aldolase is an enzyme in glycolysis but is not directly linked to sodium pump dysfunction and hemolysis as described in the case of pyruvate kinase deficiency. So, only one that made sense here is pyruvate kinase. So, let's move on to the next question. We have a 12-year-old male with a history of recurrent staph infections who undergoes a dihydroroorhodamine test confirming the diagnosis. The deficient enzyme in this patient acts on which of the following as a substrate? All right, I think I gave you guys enough time. If you need more time, pause the video now. If not, moving on. The patient's diagnosis of chronic granulomatous disease, CGD, indicates a deficiency in NADPH oxidase, an enzyme that acts on molecular oxygen. NADPH oxidase is crucial for generating reactive oxygen species during the respiratory burst, which helps in killing certain pathogens. Therefore, the correct answer is C, molecular oxygen. Moving on to the next question. Or moving on to the reasoning why the other answer choices are incorrect, my bad. A, hydrogen peroxide. NADPH oxidase produces superoxide anions, which can be converted into hydrogen peroxide. However, the primary substrate is molecular oxygen. B, nitroblue tetrazolium. The nitroblue tetrazolium assay is now considered obsolete for diagnosing CGD. Dihydrohodamine is a more accurate test. Nitroblue tetrazolium was a wrong answer on a previous NBME, emphasizing the importance of dihydrohodamine. D, halide hydroxyl radicals. Myeloperoxidase, not NADPH oxidase, is involved in the generation of halide hydroxyl radicals. Bleach, hypochlorous acid. E, NADPH. NADPH is a coenzyme used in various reactions, including those produced via the glucose-6-Phosphate dehydrogenase pathway, but is not the substrate for NADPH oxidase. Now, let's move on to our final NBME-based application reasoning question. We have a 4-month-old boy that presents with a 2-month history of frequent crying and tremulousness occurring a few hours after feeds. Examination reveals massive hepatomegaly, hypoglycemia, and lactic acidosis. Which enzyme is most likely deficient in this patient? Pretty straightforward one. Alright, I think I gave you guys enough time here. If you need more time, pause the video. Otherwise, moving on. The presented symptoms, including the hepatomegaly, hypoglycemia, and lactic acidosis, are indicative of Von-Gerke disease, a glycogen storage disease type 1. This disorder is characterized by a deficiency in glucose-6-phosphatase, an enzyme essential for the release of free glucose from glycogen. Therefore, the correct answer is C, glucose-6-phosphatase. Let's go down and break down why the other answer choices are incorrect. A, glycogen synthase. Glycogen synthase is involved in the synthesis of glycogen, not its breakdown. B, glycogen phosphorylase. Glycogen phosphorylase is involved in glycogenolysis, but is not deficient in Von-Gerke disease. D, debranching enzyme, amylo-1,6-glucosidase. This enzyme is responsible for removing branches in glycogen, not deficient in Von-Gerke disease. E, branching enzyme, amylo-1,4-2,1,6-transglycosylase. Branching enzyme is involved in glycogen synthesis, not its breakdown as seen in Von-Gerke disease. So that will do it for our review on G6PD deficiency. If you have any questions, comments, or concerns, please feel free to leave them down in the comment section below. If not, this is FixerMed signing out. Have a good day, everyone. Goodbye.